All right, I'm having a real time with this particular recording. There are a lot of ambient house noises. The dryer's going, the washer's going, dishes are being done, and the dogs are growling. So, and it's not my first time trying to record this particular video. So, really crossing my fingers.
Chapter 4, tissues. Tissues are arguably the most important topic, the most important chapter that we need to cover in all of A&P, because tissues never go away. They come back to haunt you over and over and over again. They will come up in every single chapter. So it's critical that you master tissues as soon as possible. If you don't master tissues early on, or I hope this isn't the case, but ever, then you'll struggle in the rest of 241. But even worse, you'll continue to struggle in 242 because, frankly, any A&P instructor worth their salt should be emphasizing tissues throughout A&P. It's like the thread that weaves all of A&P together, okay? And a tissue, in order to qualify as a tissue, it has to meet a few conditions. It has to be a collection of cells, so not just one cell, okay, a collection of cells that have the same function or complementary functions or overlapping functions, and that collection is physically separated from any other such collections. Okay, it's a collection of cells, same or overlapping function, and physically separated, delineated from any other tissues. Okay? Histology, by the way, is the study of tissues. And if you find you're really struggling with tissues, like all of the materials that I provide are not cutting it for you, then instead of turning to Google, which can be very dangerous, I would recommend that you find maybe an older edition of a histology book. And a lot of histology books are spiral bound. They have a lot of images in them, and they can help provide you with extra practice. But it's absolutely not required that you get a histology book. I wouldn't even say it's recommended. You should find plenty of practice on Canvas amongst all the different materials that I provide. Okay. There are 4 basic types of tissue in animals, and those are epithelial tissues, connective tissues, muscle tissue, and nervous tissue. Okay? In the next slide, which is 3, the third slide here, just kind of an overview of some of the different sort of locations where you might find these tissues. However, I find this image to be a little bit misleading because, by definition, an organ, actually, let's use a different symbol, is guaranteed to be comprised of at least 2 different types of tissue. Okay, so pointing at a bone and saying that a bone is comprised of connective tissue, yes, it is, but it also features other tissues, and for that matter, different types of connective tissues. Okay, so be careful about that, and I think that will also come back to haunt you, or at least that rule of thumb can be helpful. Okay. Before we start studying the different types of tissue, let's talk about how we prepare a tissue slide on the 4th slide here. In order to prepare a tissue sample for examination through a compound light microscope first we have to preserve the sample, preserve the tissue, because if we don't preserve it, it will start to decay and therefore stop expressing the different characteristics that we're hoping to see, okay? So first priority is to get that preserved, okay? Then we slice, and we slice so thinly that the sample allows light to penetrate through it, okay? Now, sometimes that slice is a little thicker. Sometimes that slice is a little thinner. Sometimes that slice happens to be really loaded with a lot of features. And sometimes that slice happens to be not highly loaded with a lot of features. And that's one of the many reasons why a tissue in one slide may look quite distinct from the same type of tissue on another slide. What that means is that you have to practice identifying your tissues by sight. Because one example, one specimen may not quite look like the next. All right? The next step in preparing a slide is to stain, apply a dye, or sometimes more than one dye or stain, okay? And that's another variable that can lead to two specimens, two samples of the same type of tissue or the same organ for that matter, looking rather different, because one preparer may use this stain and another preparer may use no stain and another preparer may use a different stain, okay? One preparer may use a lot of stain, another preparer may use very little stain, and so the saturation can absolutely vary from one specimen to the next, okay? You do need to know the steps of preparing a tissue sample for a microscope slide, okay? All right, now let's start talking about epithelia, the epithelial tissues. And boy, do we need to know a lot of epithelial tissues. We also need to know a lot of connective tissues. Thankfully, there are only 3 muscle tissues, and nervous tissue is nervous tissue is nervous tissue. So nervous tissue is probably your favorite. Okay. All epithelia are comprised, oh, by the way, epithelium is singular. Okay. Epithelia is plural. Okay. All epithelia are comprised of sheets. Some epithelia are comprised of a single sheet. Some epithelia are comprised of multiple sheets, okay? But always sheets of cells, all right? And these sheets either coat or line or cover different parts, different structures in the body. Some epithelia are associated with glands, but that glandular epithelium we're going to save for much later in this same lecture, okay? In fact, by the time we get there, you will have forgotten that we were supposed to talk about that. All right, and for now, we'll focus on those that coat and line and cover rather than comprising glands, all right? One of the factors or features of the different kinds of tissues that you want to master are their functions, okay? Because if you can identify a tissue on or in, I should say, a micrograph, a photograph that's taken through a microscope, and you know what that tissue does, then sometimes you can figure out what organ you're looking at, okay? Here's a list of the general major functions of epithelia, but it's probably better for you to learn functions of specific epithelial types, okay? So let's move on now to those covering and lining epithelia. I'm looking at what I want to say is slide 6. I'm actually going to skip slide 7 for now. We're going to use it as an overview or a review at the end instead of at the beginning. Okay? And now I'm on slide 8, which talks about polarity. All epithelia feature polarity, which makes it sound like they're charged, which isn't necessarily wrong. A lot of cell structures and even cell surfaces will feature a charge, and overall the body is charged. We have an overall negative charge, and certainly we want charges to be separated where some cells, like neurons, are concerned. But here, what polarity refers to is the fact that the two faces of an epithelial look different. They have different forms and different functions, and that's hopefully very sensible to you. Hopefully you learned long ago that a major dogma in biology and science in general is that form dictates function, right? So if these two faces, these two surfaces, they look different, they're anatomically distinct, then it would make sense that they would also be functionally distinct, okay? And we're going to use this really, really oversimplified image. Up here at the top. Here is our sheet. In this case, just one sheet, okay, of epithelial cells. Just a single sheet this time, all right? And there's this surface versus this surface. I think, if I remember correctly, we already learned the term lumen. Right, that space, that internal space, whenever we're looking at a tube-like or a bag-like organ, and boy, do we have so many of those. So many. Well, certainly, and I'll change colors here just to make this viewable. Such an organ is very likely lined with epithelium, okay? There's one of the surfaces of that sheet, and there's the other surface of that sheet, okay? Well, in the case of a tube-like or sac-like organ, the lumen-most surface is the apical surface, okay? Whereas if we're looking at the epithelia that comprise the epidermis of our skin, the apical-most surface is, well, frankly, what I'm looking at. Right? Whenever I look at my skin, I'm looking at the apical-most surface of my epidermis, okay? Whereas this surface is called the basal surface, okay? That apical surface is most exposed. It's either exposed to a lumen, a cavity, right? Or a surface, or maybe even a coelom, okay? Most apical surfaces are really, really smooth, but some are folded. And those folds may be expressed as a microvilli. And in fact, in this image at the bottom of the same slide, this is apical surface. This is basal surface. Again, a really, really simple epithelium, just one sheet. But here, these teeny tiny finger-like folds that are just expressed on the apical surface, those are microvilli. Be very, very careful. Microvilli are so small, right? They're subcellular. They're so small that they cannot be seen through a compound light microscope. We would need an electron microscope. Okay. That basal surface is in turn attached to almost like a sticker or a sheet of adhesive that's called basal lamina. Basal lamina. And this is where things start to get really, really wicked. This layer, maybe we should use a different color. Maybe like this guy. This layer here. And we'll talk about this on another slide, but it hurts, so we might as well start that ball rolling. That layer that underlies the epithelium, okay, it's called the basement membrane. The basement membrane. And the topmost layer, I know, shut up, Tessa, this is getting horrible, of the basement membrane is the basal lamina. What that means is that basal surface, basal lamina, and basement membrane are not the same thing. Isn't that horrible? Ew. Basal surface is a feature of the epithelium, whereas basement membrane is something that underlies that epithelial sheet, okay? And its job, the basement membrane, is to anchor the epithelium, but also provide strength because a good chunk of it's made out of connective tissue. I will come back to that automatically, okay? Polarity, again, here refers to having two anatomically distinct surfaces and therefore two functionally distinct surfaces for every epithelium. Another sort of guaranteed feature of any epithelial tissue is intercellular junctions or specialized intercellular junctions. And when I was you, I learned intercellular junctions, so that's what comes out of my mouth most often. Okay. Animals, really by definition, feature three intercellular or special intercellular junctions. They are called gap junctions, tight junctions, and desmosomes. Okay? That doesn't mean that only animals feature specialized internalized junctions. It means that only animals feature these specialized intercellular junctions, whereas a plant, for instance, features plasmodesmata. Animals never feature plasmodesmata. Okay? Well, you may have already studied gap junctions, tight junctions, desmosomes in, for instance, 160, or maybe you've taken 212 before. But just in case you're rusty or you never felt great about them, I'm going to share a really detailed, kind of ridiculous analogy with you, okay? So think about the mall. I know, we haven't been there in a while, have we? Suppose that the mall happens to have two storefronts side by side open for vacancy at the same time. Okay, so two adjacent storefronts simultaneously available. All right. Well, Gap, which I'm choosing on purpose, the company Gap Jeans, you know, they see this opportunity and they grab it. Okay. And what they do is they set this storefront up as Gap, like as in adult Gap, okay? And they set this storefront up as Gap Kids, okay? And they get permission from the owner of the mall, okay? Ooh, I don't know if you could see me do this. Maybe I'll do it just in a different color. I don't know how to show that. I'll use the yellow. All right. They get permission to tear down this wall in between the two. Okay? It turns out it's not a structural wall. And what they do, gosh, I hope this translates, is they hang two really sturdy vinyl, burlap, something sturdy curtains, okay, side by side, and they stitch a really cool pattern through there. So the pattern kind of looks like a negative of itself depending on which side of the curtain you're on, okay? Those two curtains are now stitched together, all right? Here, this is just open space, like an archway almost, all right? And here, obviously, the wall is still intact, okay? And that wall still being intact, they take advantage of that. Let's see. What they do is they put the return policy, which is the same because it's the same company, okay, up on this wall. And then because it's a shared wall, they just put bolts all the way through to anchor those return policies to the wall. Okay? Well, in this analogy, this bolt business these are desmosomes. Okay? And this stitching business between these two curtains, those are tight junctions, right? If I stitch through two sheets and pull tight, there isn't a whole heck of a lot of space in between. Tightly bound, tight stitching, okay? And this walkway, which from a marketing standpoint is brilliant, because once you've enticed the customer into Gap, you can entice them into Gap Kids from within, okay? This represents a Gap. Oh, that's why she chose Gap. A Gap junction. In other words, a Gap junction is like a pore or a portal. And these two storefronts represent cells. Adjacent cells, okay? Adjacent epithelial cells have desmosomes, gap junctions, tight junctions in place. Now, that doesn't mean that only epithelial cells can have these. For instance, there are lots of desmosomes and gap junctions in place between adjacent cardiac muscle cells, okay? What we're saying is that all epithelia feature these specialized intercellular junctions. Do they all feature them in the same ratio? No. Some epithelia may be loaded with tight junctions, much more so than the other two types of junctions. Some may be loaded with desmosomes and or gap junctions, and not so many tight junctions, okay? So the ratio of these can absolutely vary, but it's always a safe assumption that Epithelia will feature these, okay? Moving on, slide 10, I believe. Well, this we already covered, which is nice. So we know that all epithelia feature polarity. We know that all epithelia feature specialized intercellular junctions, and we also already know that all epithelia feature a basement membrane, okay? And because it is really tough, I've given us this little schematic up at the top. Basal surface is an aspect of the epithelium itself, right? Whereas these two laminae, they together make up the basement membrane. The basal lamina is the portion of the basement membrane that is closest to the basal surface, and the reticular lamina is the portion that is furthest from that basal surface, okay? The basal lamina acts as like adhesive, okay? And the reticular lamina, because it's comprised of connective tissue, is, or at least connective tissue fibers, is conveying strength, okay? One of the reasons why it's difficult to erode, at least many of our epithelia, is because that basement membrane that underlies them is strong, okay? Here, we can get a better appreciation, because a lot of students miss this. Is basement membrane very thick relative to epithelia? No, it's actually so thin, you're never going to see it through a compound light microscope. It's really, really thin. So, for instance, if you were ever to identify a layer as a basement membrane, like on an exam or a quiz, I would know instantly that you didn't understand the concept of basement membrane. It's very, very, very thin, okay? Let's move on to the next slide, okay? We already established that basement membrane is comprised of basal lamina and reticular lamina. Well, that reticular lamina, again, is comprised of connective tissue. And all connective tissues have fibers, or at least fiber ingredients in them. And it's those fibers, especially collagen fibers, that lend support to the very thin, but very important basement membrane, okay? Next slide, which is 11. All epithelia, and this is really going to be handy for you if you can remember it, are avascular. They do not feature blood vessels. Avascular. But they are innervated. They do feature nerve endings. So blood vessels, no. Nerve endings, yes. Okay? Let's go to the next slide. All epithelia are capable of regeneration. In fact, I would say that epithelia have a superpower, and that it's regeneration. In that, if you were to remove an epithelium entirely, it would regenerate. It would reform and look the same as it did originally and function the same as it did originally, okay? Which is just wonderful. We really benefit from that, okay? Now, I'm going to go back to that slide that was early on that I said we were going to treat as a review. Just a little review. We already talked about polarity, meaning that epithelia have both an apical and a basal surface, specialized intercellular junctions, gap junctions, tight junctions, desmosomes, a basement membrane. We know that epithelia are innervated, supplied with nerve endings, but that they lack blood vessels, which usually means they have adjacent life support in the form of those underlying connective tissues. By the way, I'm on slide 7. Oops, I did not mean to write that. But, all right, and that epithelia can, will, and do regenerate, that they have that superpower, if you will. We also talked about sort of the naming principles of epithelia. So I'm on slide 14 right now, where at least for the most epithelia will have a word, a descriptor that tells us how many cell layers, and we'll have a word or descriptor that tells us the shape of the cells, at least apically, which is what this little note here is for. Anywho, we did already talk about simple squamous epithelium being the thinnest stuff we have. From the side, if you will, we would see those sheets as very thin lines, so thin, in fact, that this thin line is actually 2 separate sheets of simple squamous epithelium, okay? But if we were standing on top of simple squamous epithelium, it would look a bit like someone had cracked too many eggs into a frying pan simultaneously. We also expect to find simple squamous epithelium in the serosae. In other words, serosae are made out of simple squamous epithelium, lining blood vessels, lining the heart, associated with the kidney. The renal corpuscle is comprised of simple squamous epithelium. And the alveoli, also known as the air sacs of the lungs. We already talked about simple cuboidal epithelium. I'm on slide 20. And often the sort of poster child for this tissue is going to be the kidney, because a section through the kidney will render lots and lots and lots of almost bracelet-looking configurations, all made out of simple cuboidal epithelium. Okay, here we have plenty of cytoplasm to go around, so this is a much more appropriate tissue for secretion and absorption. Whereas, backing up 2 slides, simple squamous epithelium is much more appropriate for filtration via diffusion. Okay, that doesn't mean it can't secrete, can't absorb. It just means that it's not likely to. Back to slide 20. Again, we expect to find simple cuboidal epithelium, specifically in the renal tubules of nephrons, whereas the renal corpuscle, what we drew and mentioned and compared to a snake head with a ball of yarn in its mouth, is simple squamous epithelium. Okay, and then also any small glands like sweat glands, for instance, will be comprised, at least in part, of simple cuboidal epithelium. So those are the 2 locales that I would have you emphasize, is renal tubules and small glands. Moving on to simple columnar epithelium. Simple columnar epithelium, I feel, looks like somebody has a whole bunch of books. Maybe there are different volumes of a set of encyclopedia, and therefore they're all the same height. And because they're all the same height, the nuclei within, they end up to be more or less aligned with each other. And the poster child for this particular tissue is the small intestine, which your section may or may not have already discussed. Chances are we did. We hopefully already learned that the small intestine is not just a water slide. It's not just a chute that we send foodstuffs through rapidly. We don't want foodstuffs to go through rapidly because then we won't have enough time to apply digestive enzymes, nor will we have enough time to absorb nutrient monomers. So rather than being a sort of water slide, if you will, the wall of the small intestine, slide 22, by the way, it's folded. These are called circular folds. And then that surface is folded. These folds are called villi. And then that surface is even folded. And those are called microvilli. Just to maximize the surface area that's available for both secretion and absorption. And if we zoom in on just one of the villi, which would be a villus, okay, then we would better see, oh yeah, that villus has sort of a coating, if you will, that's made out of simple columnar epithelium. Okay, so all these little cells here that I'm doodling in are simple columnar epithelia. And so what we're seeing in the photo here is two villi side by side. Here's a villus, just the edge of a villus, and here's a villus, just the edge of a villus. So in other words, if I were to add another villus next door to the one that I've already drawn, the photo was taken sort of right in the interface between those two villi. Often, simple columnar epithelia will, I guess I shouldn't say often, it's very plausible for simple columnar epithelia to feature microvilli, and certainly in the small intestine, this epithelium does feature microvilli, but microvilli aren't as sort of guaranteed to be featured by this epithelium as, say, cilia would be to be featured by pseudostratified columnar epithelium. Okay. Plenty of cytoplasm to go around here. So again, absorption and secretion, more so than diffusion. Okay, and we're expecting to find simple columnar epithelium in the stomach, and in the stomach it would not feature microvilli. In the small intestine, obviously, and that's actually what this photograph is of, right? In the large intestine, but in the large intestine, no microvilli. And in the rectum, which is actually part of the large intestine. It's just like our neighborhood of the large intestine. Okay. Some glands will feature simple columnar epithelium. The gallbladder features simple columnar epithelium. It is possible for simple columnar epithelium to feature cilia, but we only see that in the very sort of distal, smallest respiratory tubes, in the fallopian tubes or oviducts, and in some parts of the uterus. So it's very uncommon. It's much more common to find cilia associated with pseudostratified columnar epithelia. Okay, and I think that's our next slide, actually. Yeah, so slide 24, pseudostratified columnar epithelium. About 95% of the time in the body, this particular epithelium will feature cilia apically. And those cilia can be very dense and long, but they also can be relatively sparse and short. So no guarantee that you're going to have this really lush carpet that you see in this particular micrograph. This happens to be the trachea, and that tends to be kind of the poster child for this particular tissue. We can also find pseudostratified columnar epithelium in sort of the early branches off of the trachea. So the bronchi, right? And then I think we already established that we can find pseudostratified columnar epithelium in the nasal cavity as well, and in part of the pharynx. We talked about that in our last lecture or lectures. We do feature pseudostratified columnar epithelia in some of the ducting of the male reproductive system. And in that particular locale, we're not going to see cilia. So that's sort of like the weird exception to ciliation in pseudostratified columnar epithelia, because there's plenty of cytoplasm to go around. Secretion is certainly a biggie. However, since cilia are so common, it's a little more difficult to get absorption done because cilia don't provide more surface area for absorption. It's more like they provide more obstacles that would prevent absorption from being maximally efficient. And those cilia are typically in place where they are present to help sweep mucus, to propel, if you will, mucus. So, for instance, in the trachea, we're constantly secreting mucus via modified, specialized, slightly different, further differentiated, if you like that, cells that are called goblet cells. Goblet cells make mucus, and they send mucus to that apical surface or to the lumen of, in this case, the trachea. And if we did not have cilia, then that mucus would eventually end up in our alveoli, which is not an option. So those cilia instead sweep that mucus, and hopefully any debris gets trapped in the mucus. It's sort of like the body's fly trap, and send that mucus, sweep it up, sort of like an escalator, to the pharynx. And then the pharynx is connected to both your trachea and your esophagus. So once that mucus comes up to your pharynx, you then swallow it and send it down your esophagus to your stomach or your pit of despair. And you do that automatically all day long. That's a chief and normal sort of behavior or function. Now, be careful with goblet cells, because goblet cells are also often found in simple columnar epithelium. So I'm backing up to slide 22. Here is another goblet cell. Okay, and then there's another one right here. Wow, that's the worst star I've ever made. If it's any consolation, the student's texting me right now, so I'm getting distracted. Okay, anywho, goblet cells again make mucus, and we don't just or strictly name mucus in pseudostratified columnar epithelium. So you want to think of goblet cells as modified columnar cells, not necessarily modified pseudostratified columnar cells. Okay. Moving back to slide 24, one way to differentiate the pseudostratified columnar epithelium from the simple columnar epithelium is to really notice that even though these are sort of brick-like cells or book-like cells, they don't all achieve the same height. And so their nuclei end up all over the place. And I feel the illustrator's done a good job here, really highlighting that. So much less alignment among the nuclei than what we saw in simple columnar epithelium. Moving on, I'm on slide 27. And slide 27 is going to be especially important for you because in the file called Practice Your Tissues, there is one tissue that is not represented. And this is the one. This image is an image of non-keratinized stratified squamous epithelium. And hopefully I've already emphasized in class that all 4 words are necessary. We also need to be able to identify keratinized stratified squamous epithelium. Okay? The non-keratinized stratified squamous epithelium, we actually probably already know where to find it, since we've already talked about mucosae and serosae quite a bit this quarter. The non-keratinized stratified squamous epithelium, oral cavity, part of the pharynx, lining the esophagus, which is actually where this photo was taken. The vagina, the anal canal. Okay, whereas keratinized stratified squamous epithelium, that makes up the epidermis. Okay, and we might as well use this image to talk about the difference between keratinized and non-keratinized stratified squamous epithelium. Notice that in this particular micrograph, which is just a photograph taken through a microscope, even these outermost layers feature nuclei. Okay? In other words, all of the cells in these strata or layers are living. All right? Whereas if we were looking at non-keratinized stratified squamous epithelium, roughly half of the cells would feature conspicuous, obvious nuclei, and then roughly half, those are that are apical most, would not, and they would look quite flaky. There would be actually a substantial layer, about half, that would just look like sort of like layers in a croissant, just very, very flaky. Okay, and the reason why that outermost layer in keratinized stratified squamous epithelium looks flaky and doesn't feature conspicuous nuclei is because those outermost or apical most cells are dead. Okay, in part because they're far from life support, and in part because they're full, like overrun with keratin. They're so full of keratin that there's no room for cytoplasm, there's no room for organelles. And we would see living cells comprising just sort of the basal half-ish of epidermis in this particular case, okay? And there is a lovely image in your Practice Your Tissues file of keratinized stratified squamous epithelium, but also the next chapter is skin, so we'll see that many times over. So if you want to refer to that now, it's not going to hurt you, certainly. Because this is a stratified epithelium, its chief function is protection. So to protect from abrasion, right? If it's got multiple layers, then I can afford to lose some of those layers. All right, and that's the case certainly in both non-keratinized and keratinized stratified squamous epithelium. But when we talk more extensively about keratinized stratified squamous epithelium being the epidermis, we'll also identify that, well, having keratin means that you've waterproofed that apical most chunk, and therefore we're protecting not just from abrasion but also from desiccation. Okay. let's see. And then I've just highlighted for you, hey, don't forget, this helps to make mucosae. If we back up to slide 22, I've also highlighted in red mucosae, okay? Anywho, moving on to slide 29. Oh, I think. Did I skip one? No, I didn't. Okay, 29. Transitional epithelium. So, doesn't really adhere to the naming rules or the classification rules of the other epithelia. There's no indication of how many layers. There's no indication of sort of the shape of the cells, right? But this is actually considered a stratified epithelium. The reason why there's no cell shape name or descriptor included is that the shape of these cells tends to change depending on what the organ is doing. And that's one reason why this might be called transitional epithelium. Another way to sort of keep that word transitional in mind is if you look from apical to basal poles, if you will, you'll notice that the shape of cells changes dramatically. So much so that by the time you're at the basal surface, some of the cells are looking columnar, right? And whereas when you start at the apical surface, depending on what the organ was doing, they look almost like really puffy, oversized squamous cells, or I think they look like muffin tops. Okay, and this particular micrograph was taken in the urinary bladder, which is one of the poster children for transitional epithelium. When the bladder is relaxed, I'm more likely to see these muffin tops. Whereas when it's stretched because it's full, in other words, when it's distended with urine, then the shape of these cells changes and isn't as muffin toppy. Okay, so be careful with that. And also be aware that not only do we find transitional epithelia in the urinary bladder, but also in the ureters, which are the tubes that run from kidney to urinary bladder, and in the urethra, or at least part of the urethra, which is the tube that eliminates urine from the body. Okay, so in terms of functions, you might think of this as being a functionally transitional epithelium because it's ready to stretch, it's ready to change. Now, we do in the human body feature also stratified cuboidal and stratified columnar epithelia. However, they are very rare, which means I'm not going to ask you to be able to recognize them through a microscope or even in a photograph, okay? Just know that we do feature them, and that stratified cuboidal epithelium, it's featured in some of our glands, and it's stratified based on a really sort of thin line. It's 2 cell layers thick rather than 1. Okay, so technically stratified, but still quite thin. Okay, and stratified columnar epithelium we can find in sort of patches in some glands, in part of the male urethra, and part of the pharynx, typically found wherever we're transitioning from one epithelium to another, okay? And it's only the apicalmost layer of stratified columnar epithelium that's columnar in appearance, but hopefully we were expecting that since that's really one of the rules for that shape descriptor. And then don't really worry about finding photos because, again, I'm not going to ask you to recognize them, okay? Now, the next portion of this file, which is 4A, is about glandular epithelia, which is typically very difficult to wrap your head around. Everybody struggles with this particular topic. So I'll lecture here about it. I'll cover it in this sort of electronic form of a lecture. But I'll also come back to it in person, just in case, because I don't want anybody walking away still with an excuse to struggle. Okay. So technically a gland is anything that makes up a secretion, okay? Now be careful with the terms secretion and excretion. Excretion means get it out. To excrete means to get out, okay? And certainly we can secrete to get out, but we're not strictly using secretion to get rid of things.
Sorry for interrupting. I have to skip less than a minute of me talking gibberish. Oh no, I didn't get that right. Almost. Come on. Takes me longer to skip it than just let it go. Oh. Such a hair trigger.
You done?
No.
You almost?
No, you're being recorded right now.
Okay, so excretion is always going to include some secretion, but not all secretion is to the end or toward the goal of excretion. Okay. Anywho, we have two major sort of types or classes of glands. We have endocrine glands and exocrine glands, and we need to know all the differences that they typically feature. Endocrine glands secrete their products internally, and those products are meant to stay inside the body. They're not meant to be excreted. They're not meant to be gotten rid of, if you will. And those products are called hormones. By definition, a hormone is a chemical signal that travels via the bloodstream. And so endocrine glands secrete their products into the bloodstream. Exocrine glands, they secrete their products externally. They're sending their products to the outside of the body. Okay, and a great example of an exocrine gland would be a sweat gland. Another example would be a sebaceous gland. We can potentially have unicellular glands, believe it or not, and we can have multicellular glands. Well, endocrine glands are all multicellular, whereas exocrine glands can be either unicellular or multicellular. The unicellular version, if you will, of an exocrine gland is a goblet cell, and we met those recently when we were looking at modified columnar cells. Whereas an example of a multicellular exocrine gland, a sweat gland, a salivary gland, a sebaceous gland, for instance. Okay, I'm now on the next slide, which is 33. Endocrine glands do not have ducts. There's no duct, no tube through which secretions travel to be released, okay? Instead, we're going to release into surrounding fluid, and then those products are picked up from that fluid by blood, by the circulatory system. And surrounding fluid actually has a number of names, but this is probably the first time we're encountering any of those names. So interstitial fluid, surrounding fluid, or the fluid in between tissues or in between cells even, is sometimes called interstitial fluid. Okay. Anywho, hormones, again, are the specific products of endocrine glands. They are, again, signals, chemical signals or chemical messages, if you want, that specifically travel through the blood, okay? And hormones target different organs. Typically, a hormone does not target every single organ or every single cell in the body. But there certainly are some hormones that have many, many targets. Exocrine glands may have a duct. It's only the multicellular exocrine glands that will feature a duct, whereas unicellular will be ductless. And I'm on slide 34 right now, by the way. Okay. This time around, we're going to send our secretions rather than into interstitial fluid onto surfaces. So into body cavities, onto skin, that sort of thing. We have far more exocrine glands than we do endocrine glands. Some of our exocrine glands are quite small. Some of our endocrine glands are quite large. Now, that doesn't mean that our exocrine glands can't also be large, but they're a little more variable in size, okay? And examples of exocrine glands include mucus glands, for instance, goblet cells, sweat glands, sebaceous glands, salivary glands, okay?
Sorry, I have to do that jump ahead trick one more time.
Unicellular, and the go-to example of that would be a goblet cell. The function of a goblet cell is to produce mucus, and a goblet cell is simply a modified, tweaked columnar cell. Okay, we're going to find these in the small intestine, the large intestine, the trachea, the bronchi, okay, secreting mucus to, depending on where, either act as a trap, like a fly trap, trapping debris, trapping potential pathogens, trapping potential irritants like pollen, or acting as a lubricant. So much more of a lubricant in the intestines, much more of a fly trap, if you will, in the respiratory system. Okay. Slide 36 has a colorized micrograph of a goblet cell, but tons and tons of vesicles crammed into the cell body of a goblet cell, and each vesicle is housing a whole bunch of mucin. When mucin comes into contact with water, it forms mucus, okay? And that's why these cells so often look swollen, because they're swollen with vesicles. Vesicles are almost like packages or bubbles inside a cell that are used to package things. So if I produce hormones in a particular cell, then I'm going to probably package those up in vesicles, at least while they're still in the cell. Or if I produce neurotransmitters inside a cell, I'm going to package those up in vesicles. Vesicles are hopefully old news from 160. And I say hopefully because, boy, are they going to come up often in this class. Multicellular exocrine glands, again, sweat glands, salivary glands, right? Even you could consider the glands of the stomach to be exocrine glands. We will see a duct and a secretory unit. A secretory unit. So if we have a really basic gland, okay, made up of multiple cells. Unfortunately, you're not here to say, we get it. Okay, multiple cells. Then this portion here, this would be the secretory unit. Okay, and this portion, the duct, often, but not always, comprised of simple cuboidal epithelia. Together, secretory unit plus duct equals the whole gland. Okay, that's important to keep in mind, especially because this is about to get a little bit crazy. Okay, we're about to meet glands that are classified in accordance to the shape of their secretory units, as well as the sort of branching efforts that both ducts and secretory units make. And the best way to wrap your head around this is to go visual ASAP. So we're going to go to slide 39, which is this is a tough one. This is quite the hurdle for many people. So when we're describing a multicellular exocrine gland, in other words, this only applies to multicellular exocrine glands, we're going to use the descriptors tubular and alveolar to only describe the secretory unit, the part of the gland that actually is making the product, the secretion, okay? And the illustrator has made all of the secretory units on this chart red, okay? Well, notice that this secretory unit doesn't branch, whereas this secretory unit, that red, does branch. It's lobed, in other words. Here, that red does branch. It's lobed, okay? Well, whenever a secretory unit branches, we say branched. That almost makes sense. Okay? However, ducts in this particular chart are depicted using yellow. And because we've already spent the term branched or unbranched on secretory units, we need a different way to describe splitting, forking in the road of ducts. And so for ducts, we say either simple or compound. Okay? So this the very first gland that's depicted would be a simple, unbranched, sorry, that's not explicitly shown already, tubular gland. And this second would be a simple, the yellow doesn't branch, branched, tubular gland. Okay? I'm going to come down the page now. Okay, this first alveolar gland would be simple, unbranched. Okay, and the second would be a simple branched alveolar, where branched, in both of these cases, refers to the branching of the secretory unit, not the branching of the duct, because the duct doesn't branch, okay? Whereas if we look at the sort of right-hand side of the slide, okay, yellow, if we trace the yellow through, yellow does branch. It's the red that doesn't. Red, separate red, separate red, separate red, separate red, okay? So in this case, this would be a compound, meaning the duct does branch, okay? Compound, unbranched, meaning the secretory unit is unbranched, tubular gland. Here, again, yellow branches. but I can't say branched because branched describes secretory units. So this is a compound, unbranched alveolar gland. And this guy here is a weirdo because some of our secretory units are nice and round, which is what alveolar means. Nice and round, okay? Whereas some of these are more elongate, in other words, tubular. Okay, making this, are you ready? Compound, unbranched, tubulo-alveolar. Crazy. So the icky part of this is that when the secretory unit branches, we say branched or unbranched. When the duct branches or splits or forks, we don't get to say branched or unbranched. We say either simple or compound. Crazy. Takes practice. Okay? Now, we can also classify multicellular exocrine glands based on how they achieve secretion. Merocrine glands, they secrete via straight up exocytosis, fusing vesicles that are inside with the plasma membrane and therefore extruding the contents of those vesicles. Okay. Holocrine glands, and again, these are descriptors of strictly multicellular exocrine glands. They actually rupture, they explode, but only the apical-most cells explode. It's not as if the gland in all the cells in that gland are going to rupture. It's just the apical-most cells that are rupturing. And because these glands are made out of epithelia, that's okay, because we're regenerating constantly. I'll show you that graphically in just a hot second. And then apocrine glands, well, if they exist, then just the very sort of tip of those apical cells will rupture, okay? But there's a lot of controversy about apocrine glands or apocrine glands because if we have them at all, the only form in the human body is mammary glands, and that's under debate. So we're really going to focus on merocrine and holocrine, okay? In the next slide, in fact, we're only shown depictions of merocrine and holocrine. Merocrine, again, we're just relying on exocytosis. Okay, holocrine, we've got regeneration, going hog wild at the basal-most surface or at the base of the secretory unit, and then apically is those most mature, oldest cells that are rupturing. Okay, and this is actually how we make sebum, which is made in sebaceous glands. Sebaceous. Okay. You might think of sebum as the oil that your skin makes. You might blame your sebaceous glands for pimples, or if you don't wash your hair for a couple days, your hair looks greasy because it's soaking up sebum. Sebum is actually not just the secretion, it's also fragments of the cells that have exploded or ruptured to extrude that sebum, which is kind of interesting, okay? Whereas examples of merocrine glands, sweat glands, are merocrine secretors, but we'll come back to that explicitly when we get to the skin. I believe that's the last slide of this lecture. It is.